In particular, what I'll talk about is microtubule associated motors. So there are two main classes of microtubule associated motors, the kinesin family of motors and the Dynein family of motors. So the microtubule is a sort of chemically polar object. One end has, the microtubule consists of a tubulin dimer. So one end has one monomer of this dimer, the other end has the other. So we call that the microtubule has a minus end and a plus end. Okay, so the kinesin are positively directed in that they walk towards the plus end of the microtubule. The dyneins are negatively directed in that they try to walk towards the negative end of the microtubule. If you look at a cargo that is carried by these motors, what's found experimentally is that the same cargo has both types of motors attached to it, both kinesins and dyneins. So if you look at some experimental tracks of cargo, these are, I think, experiments on Drosophila, you'll see that the cargoes are going, sometimes moving in one direction, then they cross over and move in the other direction, and so on. So you have teams of oppositely directed motors which are acting on the same cargo, but they give rise to directed transport. So the cell gets stuff where it needs to go eventually. So people have been trying to understand this from a theoretical perspective for a long time. But initially, a lot of the experiments were done on kinesin because it's a simpler motor to study. So what was generally assumed for a long time was that kinesins and dyneins more or less act by the same mechanism. The motor parameters that characterize the motor might be different, but the underlying mechanisms are somewhat similar. But there has been a recent spate of experiments on dyneins that show that in fact dyneins act in a very different fashion from kinesins, in particularly their unbinding behavior. So this is a set of experiments that looks at, so these, if you look at these top two graphs, these are position versus time. So you look at a cargo, you track the position of the cargo as a function of time. And what these experiments do is that at some time, they switch on a force, so you switch on an optical trap, such that the force is enough to stop the motion of the cargo. So that force is called the stall force. So you apply a force using an optical trap, which is larger than the stall force. So once you apply that force, the cargo sort of stops moving. After a while, the motors unbind and they fall off. So you plot a histogram of this residence time, the time it stays on bound before it falls off, as a function of this load that you put in the optical trap. And if you look at kinesin, so this is for kinesin, this is plus end motion, what you see is that as you increase the load, as you increase the opposing force to the cargo, the residence time sort of decreases, which is sort of what you would expect. If you pull on a cargo with a lot of force, it sort of tries to unbind, the motors try to unbind much faster. On the other hand, if you look at the same experiment for dyneins, what you see is that if you increase the opposing load, the residence time of dynein actually increases on an average, or it can increase in certain cases. So that's somewhat counterintuitive. What you're saying is that you're pulling on a cargo in the direction opposite to the direction of motion. The more you pull on it, the tighter the cargo sort of holds on. So this is what is known as a catch bond. Catch bonds are known in other biological contexts, but in the context of motor-driven transport, it's a relatively new experimental finding that has been discovered recently. So what we want to ask is that given that kinesins and dyneins act by these two very different mechanisms, the unbinding happens with very different, a catch bond in the case of a dynein, what are the implications for cooperative transport for motors which are driven both by kinesins and by dyneins? So this is the set of experiments which actually measures the unbinding rate for kinesins and for dyneins. So it does the same set of experiments. You get the histogram. From the histogram, you get the typical dissociation rates. And you plot those rates as a function of force. So what you see is that for kinesin, it's always an increasing function. So it initially increases. After it reaches the stall force, the value decreases, but again, it sort of increases with force. So this is called a slip bond. If you look at dynein, on the other hand, it increases initially until it reaches the stall force, and then it sort of decreases as you increase the opposing force. So this is the catch bond in dynein. So how does one sort of mentally have a picture of this catch bond? So the mental picture that one has is that generally when you're talking of these bond breakages and so on, what we say is that there is a barrier to this bond breakage. In the case of a normal slip bond, if I increase the force, this barrier sort of decreases with force. Therefore, the unbinding rate increases. This is the typical picture we have for a kinesin in the case of a slip bond. On the other hand, if you have a catch bond, like in dynein, the picture is that although this barrier decreases with force, just like in the kinesin case, the minimum of the potential also decreases. So effectively, what can happen is you apply force, is that the effective unbinding rate that you observe, that can actually decrease with force. And there are reasons why this happens due to conformational changes in the dynein or in other molecules which show catch bond, that as you apply a force, actually the conformation of this dynein motor itself can actually reach a more favorable configuration, which leads to lower unbinding rates. So we would like to model this type of an unbinding kinetics. And what you generally do to model the unbinding rates, if there was no kinesin, the standard slip bond is like an exponential as a function of the force. So just this, the deformation energy sets in at a force scale, which is this stall force in the case of dyneins. And then it has a functional dependence on the force. So if you pull on it too hard, then it ultimately does not change. So this is a sort of phenomenological model that we write down to sort of qualitatively explain the features that are seen in experiments. If you use this form of a dissociation rate, then what you see for a single dynein is that these red dots are the experimental data. So this is the same experimental data that I showed here. And if you use this sort of a dissociation rate, you can actually model the unbinding rate very well. Forget about these other three curves. These are to do with motors which are driven by dyneins only. This was discussed by Shudipto at the last meeting, so I won't go into that. What I want to talk about today is what happens when you have bidirectional motion. That is, you have a cargo that is carried both by dyneins and by kinesin motors. So how do we want to study this? So the basic approach is that you write down the master equation, and the master equation depends on the dissociation and the association rates. The dissociation rates are my epsilons, the association rates are my pi's, and they are different in particular for plus or for minus modes. So you have dissociation rates for kinesins, you have dissociation rates for dyneins. Same for the association attachment rates. You also have a velocity model which says that how does the velocity of the cargo change as a function of the force. These are taken from experiments. So, and one further assumption we make, at least initially, is this sort of a mean field assumption in which we say that the force that the cargo feels is equally shared between all the motors that are attached. So in that sense, the mean field analysis. Okay, so given that you have this sort of a setup, you have this sort of bidirectional motion, what do you expect? So the standard scenario in order to understand this type of behavior is this tug-of-war hypothesis. So basically it says that you have forces due to kinesins in one direction, you have forces due to dyneins in the other direction. So you have a sort of tug-of-war. And additionally, you have stochastic binding and unbinding of motors. So if you look at traces, you'll see that sometimes it moves in this direction, then maybe it stalls for a while, where it sort of jiggles around in the same position, then maybe it switches direction and so on. So this is a standard tug-of-war picture that we have for motor-driven motion where both types of motors are active. And in fact, there have been a lot of experiments, so these are actually experimental tracks, so you do see a lot of experiments which show signatures of this tug-of-war type of motion. But there is, so if you have this sort of a behavior that you have two teams of oppositely directed motors, and then if I were to somehow disable one type of motor, so if I were to disable the minus directed motors, for example, and if this was true, what I would expect is that all my cargo would now sort of zip through in the positive direction. So this is the sort of naive expectation that one would have based on this tug-of-war hypothesis, that if you have a normal cell, so this is a cartoon of a Drosophila cell, this ends of the stars are the positive direction, the inside over here is the negative direction of the microtubule. So generally you have a distribution where the motors are everywhere, so the cargo is everywhere. If you sort of somehow inhibit the function of dynein, then you would expect that everything would go and accumulate at the plus ends. If you somehow inhibit the action of kinesin, you would expect that somehow everything would cluster at the minus ends. However, when you do these experiments, very often you see that this is not what actually happens. If you inhibit the action of any one type of motor, the other type of motor also does not function. So again, this is experiments on Drosophila, where you have sort of inhibited the action of one type of motor, and what you see is that you get clumps of this cargo. So these are axonal cells in Drosophila. What you see is that there are clumps of cargo that are distributed throughout. There is no motion that takes place. So even though this naive picture of a tug-of-war does actually work in many cases, but it leads to what is known in this literature as the paradox of codependence. That if you actually stop one type of motor, the cargo as a whole stops moving. So I'll just describe one result of this analysis that we have. And so what we do is that we look at the average run length, how far a motor walks before it falls off. as a function of the unbinding rate of dynein. So this axis, as you go along this axis, what you're doing is you're weakening dynein. What you see is that in some parameter range, so there are many parameters which are chosen from biological experiments. There's only one free parameter, which is the depth of my deformation energy, which we do not have an experimental input of, but the rest are all chosen within biological limits. So what I see is that as I weaken my dynein, my average run length in the positive direction increases, which is what I would expect based on the tug-of-war scenario. However, if you now look at a different point in parameter space, so this is an attachment rate of 10 motors are attaching per second. If I now change it and look at an attachment rate of 1 motor attaching per second, and again I do the same thing, I weaken my dynein, what I see is that the average run length now decreases. So this, and basically if you look at these contour plots, so these are plots in this ratio of the detachment to attachment rates and the ratio of the stall force to the detachment force, you'll see that in this case the colors sort of go from blue to red. In this other case, the colors go from red to blue, which is that qualitatively you have two completely different types of behavior. The model is the same, but depending on where, in what part of parameter space you're looking, you can get both tug-of-war type of transport, you can also get codependent motion type of transport. And so we have analyzed this in various parameter regimes. You can also have stuff where, you know, the run length initially increases and then decreases and so on. So I'm not getting into that, so my time is up. So what I'll just end with is that, so this part I have not talked about, it was talked about last year, that unidirectional transport by catch bond shows non-monotonic velocity profiles. What we have seen in the case of bidirectional transport is that it can exhibit both tug-of-war as well as codependent transport characteristics. And also this thing which I have not shown today is that I showed data on run length. You can also characterize the motion by pause time and switch time distributions, that how long the motor pauses before it moves and so on. So those also show very non-trivial behavior in the presence of this catch bond. Currently, we are also working on improving this mean field assumption and having stochastic load sharing between motors and so on. So this is work done with Sudipto and Abhishek, both of whom are here, and Steve, who is at ICER Mohali, Anil at Pune, and Shukriyo and Palka, who are at IIT. Thank you.
So, what should be the impact of the mechanical properties of in large length scale or something for this catch bond? Is there any evidence for the, elasticity or some kind of stuff like that?
You mean experimentally?
Experimentally.
Not that I know of. See, these experiments, these are basically, a lot of them are from Roop Mallik's lab at the IFR. These are very recent experiments, and in fact, the community has only slowly been accepting the Dynein has shown a catch bond. So as far as I know, there are not too many experiments that look at this large.
The slide where you showed comparison with experimental results, there is this non-monotonic behavior which catches the effect of this catch bond.
Yes.
Then in your numerical data, I saw...
It goes and increases again.
What's...
See, that is sort of a force which they do not explore in experiments. But even naively, you can, so what we are saying is that, okay, as you're pulling on it, if you're below the stall force, the detachment rate increases. After the stall force, the detachment rate decreases. But eventually, if you pull on it with a large enough force, the motor is bound to fall off. So that regime is, those forces, if you look at the numbers, are very large forces. So those regimes are not investigated in the experiment. But naively, you would expect that would have to be. If I really pulled hard enough, it would have to ultimately come out. So that is the source of that increase. But those forces are really large. You do not really access those forces in a biological way.
And one can comment that in the due to the catch bond, the response is negative in that regime. I mean, can one say that one has negative response due to the catch bonds?
So what would you exactly mean by a negative?
Because you're just forcing it and...
Yes, so effectively you're saying that the dissociation rate is actually decreasing. So in some sense, yeah, I guess.
Maybe somewhat related question. I mean, in terms of underlying interaction potential or something, will you be able to derive the form of that detachment rate?
So that
is that just a phenomenological?
A more microscopic derivation of this potential.
Because it has a kink type. What?
Yes. So it's difficult because the Dynein structure itself is actually quite complicated. You can incorporate, so slowly you can incorporate aspects of the chemomechanical cycle. In particular, for example, in kinesins, there are models which incorporate this ATP binding process, and because these are non-equilibrium systems, they are driven by this ATP activity. So there is some amount of detail that can be put in. I'm not sure.
There are some non-analyticities.
Yeah, so that ideally that would be great, but I'm not sure we're at a position where we can actually do that realistically, even because the experimental data in those regimes.
So probably one should check even experimental data as well.
Yes, yes. So I can show you some.
Rather than it's decreasing, it's something like a plateau or something would have convenient.
In this case, depending on how fast the potential minima decreases and the barrier decreases, you can actually get plateau type behavior that it sort of increases and then it sort of roughly remains a constant. You can also have, if the potential minima does not decrease fast enough, even with this model, you can get a purely slip-like behavior. So this form of the energy that we have written down, it can capture a pure slip, it can capture a slip catch, or it can capture a slip plateau, depending on what parameter regimes you are looking at. But yes, a more microscopic derivation would ideally be what is.
Thank Mithun once more.